welcome to Edison Open House Global Healthcare 2021. In this session, we're going to be showcasing the work of ReNeuron, the leading stem cell therapy company. With me is their CFO, Michael Hunt. Michael, it's very good to have you here. Welcome.
Hi.
Hello. So just tell me, just for those who don't know ReNeuron and what it does, just tell me the basis of your business.
Yeah, so ReNeuron was established actually over 20 years ago now, primarily to exploit the therapeutic potential of stem cells. So we are a stem cell therapy business first and foremost. We've moved a little bit beyond that core technology over the years, but the business itself was founded on research into the therapeutic potential of initially neural stem cells, and now we're working primarily actually these days with retinal stem cells, looking at blindness causing diseases. So we are a cell therapy, or more specifically stem cell therapy business.
So just for people who don't know, so stem cells, you've got kind of two sorts. One can be a universal, can be anything, and you're working with ones that are specific to particular indications.
Yeah, I mean, stem cells, as you say, take on a number of different forms. They can be defined in different ways. We are working with what are known actually in the jargon as multipotent stem cells. So they have the ability to differentiate into cells associated with the organ from which those original stem cells were derived. So, for instance, in the case of our retinal stem cells, originally those stem cells were derived from retinal tissue, and we know that those stem cells have the ability to differentiate into, for instance, photoreceptors, rods and cones, the cells that give us the ability to see. So really that's the basis of our technology. It's working with stem cells, and the challenge is really how you commercialize that in terms of generating not only stem cells to the right quantity and quality standards, but also being able to generate a therapeutic product that is easy to make, ship, store, and administer.
So these are off the shelf, as it were, stem cells, rather than being stem cells that are specific to an individual.
Yes, that's a good point. Yes, we're dealing with non-patient specific stem cells, so allogeneic again in the jargon, and that means that any patient that presents with the diseases that we're targeting can be administered with our cell therapy in theory. So it doesn't require the patient to use their own cell, or for the patient's stem cells to be harvested, manipulated in some way, and then re-injected. That's the autologous approach that does have merit in certain disease conditions, but we're very much focused on the allogeneic approach.
So let's start off with your lead project, which is to do with retinitis pigmentosa. So this is a genetic condition caused by a number of different variants, but it causes progressive blindness.
Yeah, I mean, retinitis pigmentosa, again, as you say, is a collection of gene deficiencies, if you like, that collectively form the disease retinitis pigmentosa. But the end result is the same, again, as you allude to. It's a gradual loss of sight leading ultimately to blindness, and that can manifest over quite a large number of years, actually, and can initially onset when you're quite young. So we are targeting retinitis pigmentosa generally. It's a non-gene specific approach using retinal stem cells to alleviate the effects of that condition, and best case scenario to actually restore a degree of sight in these patients.
And that non-gene approach is an important distinction because, as you say, unlike lots of other conditions which we're now seeing CRISPR being applied to, actually because of the huge number of variants involved in RP, actually you have to have this more generic approach.
Yes, that's right. I mean, we are believers in the concept of a generic approach to RP as a disease, but that's not to diminish the impact of therapies that are targeting specific gene mutations, and there are already marketed therapies, or one in particular in retinitis pigmentosa that is addressing just one of those gene mutations. Obviously, the hope for us is that we can address the entirety, or a good part of the entirety of the RP population by using a cell-based approach, which won't necessarily get at the underlying gene deficiency itself, but will certainly provide a mechanism by which sight can be restored by restoration of photoreceptors at the back of the eye, or perhaps trophic mechanisms to support the retinal function in those patients.
So where are you with your studies and trials at the moment?
Yeah, so we initiated our program in RP a number of years ago, and it's currently in phase 2A development. So we have a clinical trial ongoing in the US, and we're now opening that trial up into the UK and mainland Europe as well. And the aim for us as a business, purely and simply, is to generate what we hope will be continuing encouraging phase 2A data, positive data from that ongoing study over the next 12 months or so. And we hope that will be a gateway into getting into what we hope will be a single pivotal pre-market clinical trial to take this therapy through to the market, assuming, of course, the data remains positive, as it has been so far in this open label study.
So the additional phase 2 data that was announced in January showed a stable response over the course of a year, which obviously must have been a really encouraging result for you.
Yes, those data were actually a reiteration of data we had already previously announced last year, actually. And again, this is an open label study, so we've been able to give progress updates on patients in this study over the last couple of years, actually, as it turns out. So we're reporting it in a sense as we go. And what we're seeing is, as you say, is a very nice stable response in pretty much all of the patients we've treated in terms of their ability to read letters on an eye chart, an ETDRS chart to be more specific. And that really is a measure or a surrogate for visual acuity improvements in these patients. So the key here is to show a beneficial response in the treated eye that is stable, that is maintained over time, and of course enables the patient to improve in terms of the number of letters they can read on that chart to a sufficient extent. And certainly so far, that's exactly what we're seeing in this study. And so the aim, as I said, is to expand this study into a broader group of patients over the coming months and see if we can both sustain and hopefully build further on the positive efficacy we've seen so far on that particular visual acuity measure.
So is that, and this is a really stupid question here, but is that with just a single dose, as it were? Are there thoughts about, with some patients that you could have additional doses and that might make more difference?
Yeah, it's a very good question. The data we've generated so far, and will continue to generate in this study at least, is on a single injection basis. So the treatment was always envisaged as a one-off injection of cells, and that partly speaks to the general modality of stem cell therapies. They are mostly envisaged as one-off treatments for a lasting effect. That's the essence of cell therapy in the first instance. That said, we do believe that there might be merit in re-injection after a period of time to either boost the response or reinstate it if it diminishes over time, which we're not seeing yet, but that might be the case over a longer period of time. So repeat dosing is something that's on our agenda. We currently don't have data to support that, but it's something that we're working on in the background. But for now, in terms of the ongoing clinical study, it's very much about that single injection. The one thing we have done or are doing now in the group of patients we're treating over the course of this year is to actually do a one-off injection, but at a higher dose than we've done previously. And that's really just with the aim of amplifying the efficacy signal we've already seen. And we know that going up a level in dose is safe because we have data to support that. So obviously, if we can increase the dose, we would expect that to have, all being well, an even greater efficacious effect in those patients.
Now, what about the competition? Because you said obviously there are some companies that are going down the gene editing route, but there's a company also called, is it JCell, I think, that's a possible competitor.
Yeah, I wouldn't say we are over encumbered with competitors here, which is a good or perhaps even not such a good thing. I mean, my own view is that there's plenty of scope for more than one player in this field, and certainly in the field of cell therapy more generally. And I think that the more players there are, the more mature and better understood the whole field becomes. But in terms of what we're specifically focused on at the moment, there are one or two competitors out there. J-Site, as you say, is probably our closest competitor in terms of what they're actually doing, which again is a pure cell therapy approach. The actual administration, the way they get the cells in, and what they're looking for their particular cells to do is somewhat different from our approach. But nonetheless, they're a competitor. And of course, there are gene therapy approaches as well in retinal diseases more generally at various stages of development. So that's really where the rest of the field resides at the current time. We're certainly comfortable with our position, if you like, in that milieu. We have good early stage clinical data. We're looking to build on that this year, and all being well, that will set us up very nicely for a clear run at the market based off those data in around a year's time as we move into pivotal studies. So there is competition out there. Probably there'll be more entrants as time goes on, but that's fine by us.
You're ahead of the pack.
I wouldn't say we're necessarily ahead of the pack, but we certainly have a strong position in the field generally.
So you said you started to talk about your program for next year. Can you outline for me the calendar, as it were, going forward over the next year or so? When are you going to be reporting the final phase 2 work, for instance?
Sure, yeah, we've actually been quite specific about this in our most recent presentations, especially coming off the recent fundraising we did, of which more in a second, hopefully. And really, it's about generating the data in that ongoing RP study over the course of this year. We hope to have sufficient data to be able to give an update in scientific conference during the course of this year, probably sometime in the summer as the first pass, and then later on over the course of this calendar year as those data mature further. So yeah, there is significant news flow, we hope, arising from that study as it progresses over the course of this year. And over and above that, we're also looking to develop further our exosome platform, which is, if you like, the second arm to the business, where we are working very much in collaboration with third-party pharma companies and biotech companies, looking to generate early proof of concept data with our exosomes to deliver those third-party therapeutic agents.
So exosomes are I'd call them nano bubbles. I mean, they're kind of teeny tiny little things that can transport other molecules.
Exactly. That's not a bad descriptor, to be honest, Vivienne. And what we're doing with exosomes very much stems from, the early work we did with stem cells. So we harvest our own particular exosome candidates from the stem cell lines we've been working with for many years, and then go about purifying and characterizing those exosomes and testing their potential, both as therapeutic agents in their own right, but more recently as delivery vectors for advanced therapeutic agents being developed by third parties. So that's really where the collaborative effort comes in, and we have 3 or 4 of those collaborations ongoing at the current time, and hopefully they'll yield early data during the course of this year, with at least 1 or 2 of those collaborations ongoing.
Now, you're the CFO, so I do want to come to the money in a minute, but just before we go to that, you had to stop recruitment into the Pisces stroke study, which I know must have been really difficult and disappointing because of all the diseases that one would like to make a huge difference to. Stroke is clearly right up there. So what's the status of that project now?
Yeah, so you're absolutely right. We took a decision last year to refocus the business in terms of our therapy programs, specifically on the retinal franchise. So our stroke program is still there, but it's available for licensing. So we'll pursue that in conjunction with licensees. And actually, Fosun Pharma in China is the first such example of that. Fosun licensed in both of our core therapy assets, both our neural cells and our retinal cells, in early 2019. And we're working with Fosun to develop both of those candidates as cell therapies in the Chinese market, or for the Chinese market. So stroke is definitely still alive as a program, but it's being done in a different way. It's being done as a licensing effort rather than, if you like, under our own steam. And that's purely and simply driven by our own resource constraints and what we believe will give our investors the biggest bang for their buck in the shortest period of time. And, we are still great believers in the concept of cell therapy for stroke disability, but we had to make some choices last year. And I think ultimately that will serve the business well without having to completely, kill off the stroke program. It will still find its way, we hope, to market, but it'll be in someone else's hands, and hopefully ReNeuron will get something substantial back for that as the program continues in the hands of third parties. So Fosun is the first licensee, and we hope to do other regional partnerships over time as well with that asset.
So it's full steam ahead on the RP work. When would you expect to have a product available in your wildest dreams?
Well, we've given us sort of a broad indication of that again in recent presentations. So assuming that we get positive data from the ongoing study at the back end of this year, we'd hope then to move into a pivotal study, a pre-market study with our candidate for RP. That study won't even as a pivotal study, won't need to be huge. This is an orphan indication at the end of the day. So probably a study that would take around 2 to 2.5 years to complete. So we would hope to be in a position to launch the product or apply for market authorization, perhaps during the course of 24, or perhaps more conservatively 2025. So we have a way to go yet, but certainly we can see the end game here, and that's mighty encouraging.
And what kind of cost are you thinking of?
Again, as an orphan indication, one of the reasons we chose to focus in on retina is because we can get this program moved forward swiftly without overstretching ourselves in terms of resources. So we can certainly dispose of the ongoing study under the resources we now have in terms of cash runway. And actually even funding a pivotal study is not in any way beyond our means in terms of ability to fund that study from our own resources.
You're not talking 10,000 people here, are you?
No, absolutely not. We're talking probably around 100, possibly less than 100 patients in that pivotal study, in a condition which is relatively easy to treat, even with a cell therapy approach. So it doesn't entail long hospital stays and very, very complex surgery. It's essentially a day case procedure, as it has been in the patients we've treated so far in the clinic. So it's relatively straightforward in terms of cell therapy approaches, and that's why we've chosen to focus on it, quite apart from the clinical data we've seen so far, which again gives us great encouragement. So I think we're on a pretty clear run here, and certainly we're confident that we can fund it as we need to, either under our own steam, or if there's a licensing possibility that delivers the right value back to our investors and that opportunity arises, then clearly we'll look at that. But we want to be in a position to give ourselves that optionality, either to do it ourselves, take it all the way through to market, or perhaps to license it if there's real value to be had by going down that route.
You talked about money raised last year. I think you raised 17.5 million. How do you plan to use that investment, and how long a cash runway will it give you?
So in terms of runway, in terms of where we are now, it gives us at least an 18-month runway. So it takes us way into the second half of calendar 22, which is more than sufficient to, as I say, dispose of the ongoing retinal study, which is where a core part of that money will go, and conceivably beyond that as well. The rest of the money is going to be spent primarily on driving our exosome and our IPS, our induced pluripotent stem cell platforms forward. They've been somewhat under-resourced up to this point, but now we have the wherewithal to really drive those collaborations faster and hopefully do a little bit more. Also, as I mentioned earlier in response to another of your questions, we'd like to expand the retinal franchise that we have to look at other modes of administration and to look at repeat dosing. So again, that costs money. It's going to involve generating further data clinically and possibly preclinically as well. And so there's an element of use of proceeds directed towards those endeavors as well. But the important thing is we certainly have the cash resources to do all of that now. And that gives us the real confidence that we need to drive forward swiftly, knowing that we can actually get to an end result without cashing out.
So the job of the CFO is, to hang on to the coattails of the scientists. Yes, I can see you smiling there. That's definitely what the CFOs have to do. But It's been a pretty awful year for everyone, no matter, what kind of business you're in, personally for us all, it's been dreadful. But nevertheless, even with your CFO pessimist hat on, how are you feeling about the future for ReNeuron?
Well, yeah, I mean, we had a tough 2020, like most, if not all, other businesses. In some respects, we've had to contend with arrangements that are clearly suboptimal in terms of the way that we interact as a business and the way that we interact with others. We've had to refocus the business as well. I think we would have done that regardless of COVID, but COVID perhaps was an accelerating factor that led us to do that. And we've also, it slowed our clinical trials down, undoubtedly. During the height of the first wave of the pandemic, it was certainly the case that we weren't able to treat patients for some period of time at all. And then thereafter, certainly not at the pace that we would originally have planned to. So we're kind of through that for the time being. Obviously, we're into another wave of the pandemic now, so that may or may not influence the pace of progress here. I think we're in a better place, certainly, than we were a year ago. And of course, we have the resources to get through it now, which is obviously gratifying. So I think, we've had to contend with a difficult environment, but certainly we see real light at the end of the tunnel, and all being well with vaccines being rolled out and so on, that we can actually make the progress that we had originally planned, even though perhaps it may have taken us longer than we might have originally planned to get to the same point. So yeah, it slowed us down a bit, but from a business perspective, it hasn't been fatal, but it has been difficult.
I think that's a definite note of optimism detecting in your voice there.
That's about as optimistic a note as you'll get from a CFO, I guess.
Exactly. Michael Hunt, it's been such a pleasure talking to you. I've covered ReNeuron almost all of its years, going right back to 1997, and it's great to see it prospering. Thank you so much.
Pleasure.